morphology and anatomical study of supply specimens, that is how the morphology and anatomy is used in the taxonomical problems. So here the aim of the experiment is to study the morphology and anatomy of the Lantana species. For studying the anatomy and morphology of the plant specimen supplied, we need some materials in the laboratory such as needle, blade, saphenine, methyl grain, microscopic slide, blade, blotting sheet, measuring cylinder, ethanol, methyl blue, paper, plant specimens, etc. For the morphological study observation, we have to observe the habit of the plant, that is, how the plants look like, how the stem, root, leaf, etc. Have different features, and note down the characteristic features of all the parts, that is stem, leaf, flowers, etc. Also observe the reproductive structure, that is flowers, by dissecting them under simple microscope. For the anatomical purposes, we have to cut very thin sections of each specimen, that is leaf, stem, and taken separately, and each of them is transferred to the double staining process and observed under the compound microscope and noted their anatomical characteristics. So far as the anatomy of flower is concerned, we have seen that it is an inflorescence and individual flowers are arranged in the inflorescence. So we have to go for the flower morphology under the simple microscope and observe the results and note the characteristics which have been observed in the flower section. Regarding the double staining processes, using forceps gently we transfer the sections into a small petri dish or well, and immediately after added a drop of saphenine stain to the cover of the sample. To cover the sample, let the section sit for that stain for 3 minutes. After that, the stain is removed and 70% ethanol is added and it sit for 1 minute. After that, this ethanol is removed and again 70% ethanol is added and keep the section in this 70% ethanol for another 1 to 2 minutes for complete removal of excess saphenine in the from the section. Then remove the 70% ethanol by blotting and immediately add methyl green and let the section sit for 1 minute. Then again remove the methyl green by blotting. Immediately add 95% ethanol and allow it to sit with gentle agitation for 1 minute. Then remove this ethanol and again use 95% ethanol and sit the section in 95% ethanol for another 1 to 2 minutes. And again this ethanol is removed by blotting. So after this step, gently transfer the sections to a microscopic slide and place it under compound microscope and observe the anatomical structures, anatomical characteristic features which have been seen in the sections and mark it.